Hi. I would say, Ed, we're already recording, and this will go live on YouTube. Yeah, I'm going to hand it over to Randall to do the proper
Thank you,
introduction.
Alison.
And it's wonderful to see you here.
And
I'll
hi,
be in the
Ed.
chat.
It's great to see you again. And we won't take too long to get started. I'm just going to say, of course, many of you already know who Ed Boyden is, but just in case you don't, he's a professor at MIT, and he's got so many different positions. It's impossible to mention them all. At the Media Lab, HHMI, the engineering, computer sciences, all of them. So Ed has been incredibly busy for untold years in working on developing ways for neuroscience to better collect data, analyze data, work with data. So the tool set that neuroscience uses is really the thing that Ed has always been pushing forward. Everything from optogenetics to expansion microscopy. And I hope that today Ed is going to be able to give us new insights that can drive us forward in this direction. We've sort of already started talking about this direction a little bit the last time when we had Conrad Cording here, and he was kind of giving us his view on what is fundamental to understanding the brain and how that maybe differs from some of the things that neuroscience has been doing so far, or what tends to be the trend. And we're going to be carrying on in that spirit as well, with the next talk being, for next month's talk being Chris Owen from Boxa, who will dive a little deeper on a very specific type of neurotechnology and data collection. So with all of that said, and with Ed being ready to go and all of us here, I'm going to hand it over to you, Ed. Please take it away.
Great, thank you. All right, let me do some screen sharing here. So I've prepared just about 20 minutes of slides, really to try to convey two things. One is, what are the principles underlying our goals, right? You know, we do seek what aspirationally I like to call ground truth. Can we understand the building blocks of the brain and how they work together? The history of science has shown that, you know, when feels like physics got down to quantum mechanics, right? You know, particles and how they interact, and you know, now we have computers and lasers and the internet and cell phones and so forth. Or in chemistry, it got down to molecular bonds and the periodic table, and now, of course, have so many kinds of polymers and interesting materials every day. So it's aspirational because we're obviously not there yet. But you know, what would it take to get to the ground truth for the brain? What are the building blocks? How do they interact? So our hypothesis in our group at MIT is that in between the aspiration and the era of generating the data that tell us how to cure brain diseases and how to understand brain computations, there's a necessary building block that we have to achieve, and that's technology. We have to see things and control things. And so today I just will tell you a couple of very short stories about how we're trying to see and control different things. Aspirationally, we are trying to go for ground truth. Are we there yet in all cases? Definitely not. But my hope is that we can start by talking about what we want to do, how far we've gotten so far, and what are the cutting edges of where we are at, and where might we go next. So let me try screen sharing. Is this working?
Wonderful.
Okay, let me try to move the bar of people so it's on the side, but not including the slide. Okay. Oops. My screen just went blank. I wonder what just happened. Does anyone know what causes that in Zoom? Okay, I think I saw.
I still see. We can see the slide.
I see the slide fine now. Great.
Yeah, it might
Cool.
just be a delay.
Great. So the general theme that we're pursuing is, can we see what's going on in the brain, and can we control what's going on in the brain? And can we build tools that are ideally getting down as close to ground truth as possible, the building blocks of life, how they interact, and so forth. But, and this is where a lot of the tension comes in, we have to also look at the brain at system scale. So brain cells are enormous objects, right? They can be centimeters in spatial extent in our brains, right? And they have these beautiful tree-like architectures. But the building blocks of life, biomolecules, are nanoscale. So centimeter scale, nanoscale. We're dealing with an enormous dynamic range of spatial scale if we want to understand the brain. So that's one tension. Another tension is time dynamics, right? Brain cells compute using fairly high speed, as far as biological things go, primitives, right? Electrical pulses, action potentials that last the thousandth of a second that brain cells generate in order to compute. And then chemical exchanges between brain cells at contacts called synapses. So a brain cell would have this elaborate tree-like shape. It might form 1,000 or 10,000 synapses with other brain cells. How do you see that? How do you control that? It's a really complex problem. So I made a few animations just to try to illustrate the tension that we're dealing with. So in the brain, you have these enormous brain cells that could be centimeters in spatial extent. And then if you zoom in, the wiring of the brain is nanoscale, right? The axons and dendrites, these branches, can be 100 nanometers or even thinner in diameter. And then, of course, there are 30,000 or so genes in the human genome. They encode for who knows how many gene products, proteins, other biomolecules. Those are nanoscale, and they are often organized in nanoscale precision. So these are cartoons, of course. Images like this cannot yet be taken, but they illustrate where we'd like to be, right? Again, aspirational ground truth at scale. And then time. So brain cells, of course, are computing with these millisecond timescale electrical pulses. Not shown here in this animation are chemical exchanges between brain cells. How the heck do we see and control those? So I'll just tell you 3 short stories today about how we are trying to build tools that can confront these spatial and temporal scales. Let's start with expansion microscopy. So there are many methods for nanoscale imaging. Probably many people on this call know more about this than I do, given the theme of this group. But these different techniques, like electron microscopy and super resolution microscopy, they require expensive equipment, and they really can struggle in terms of speed and cost to image large 3D objects. So in our group, we often think about doing the opposite of what people have been doing in the past. And so we've been working on ways to take brains and synthesize dense spiderweb-like meshes of swallowable polymer throughout them, basically the same kind of material that you find in baby diapers, process the tissue appropriately, add water, and as the baby diaper material swells, you'll expand the brain. So importantly, we're synthesizing this dense spiderweb-like mesh of baby diaper polymer inside brain cells and outside brain cells, in between biomolecules and around biomolecules. We're trying to really permeate the entire brain and the brain cells within very, very densely and evenly. We anchor key biomolecules to the polymer by attaching little handles that bridge the biomolecules and the polymer. We soften the brain using detergents, enzymes, or heat. We add water, and the baby diaper polymer swells, but as it swells, it pulls apart the biomolecules. So of course, this does not work on living brains. This only works on preserved brains. That's true for pretty much all nanoscale methods that you would apply to 3D brain tissue. They work on preserved brains, not living ones. So in 2015, we showed that this works. In panel B is a small piece of the mouse brain. In panel C, we've expanded it by 100 times in volume, about 4.5 times in each direction. And so we were able to physically magnify the brain. And importantly, because the spiderweb-like mesh of the baby diaper polymer is so dense and so even, it preserves nanoscale information as we do the swelling. And we showed that in a number of early studies where we validated it by comparing to earlier nanoscale imaging methods that are very accurate, but require expensive equipment, and again, can be quite slow to apply to large 3D objects like brain circuitry. Another thing we've been doing is applying a technique that we did not invent, but that others invented before us, which is color coding brain cells. So you can take genes from fluorescent that encode for fluorescent proteins from jellyfish and coral and so forth, and you can express them in combinations in the brain. And this is the Brainbow technique developed by several groups at Harvard. A little bit of background noise. I don't know if maybe somebody needs to mute, but yeah, so this is a piece of the mouse hippocampus, a part of the brain involved with many functions, including memory and emotion-related functions and other functions. We use viruses to express fluorescent proteins in the brain. If you zoom in on a small patch of the wiring of the brain, these are different axons and dendrites, well, it's blurry, right? But if you expand, this is the same field of view in the upper right as in the upper middle. If you expand the brain in an image, now you can see the individual wiring of the brain, right? So we're very excited about this idea of using some kind of color code plus expansion to enable nanoscale imaging of brain circuitry. The expansion process is pretty easy to use. This slide isn't really meant to be read, but almost 350 experimental papers and preprints have already come out to date doing expansion. It's being applied to early cancer detection, to kidney disease, to the microbiome. It really democratizes nanoimaging. Now anybody can do nanoimaging. And we trained hundreds of groups with hands-on workshops before COVID, but in summer 2020, of course, that was not possible. And so we posted online a simple photographic tutorial with rudimentary skills like how do you handle a cover slip and how to use a paintbrush and so forth. And so many people have continued to learn expansion in the time since. So to summarize this first little short story, we developed a way to physically magnify brains and other biological systems, not living, but preserved. It's very easy to do. It's already being used in many, many hundreds of research groups, maybe over 1,000 research groups at this point. And yeah, one hope is that we can really make in the near future maps of brain circuitry with enough resolution that maybe we could try to do things like, perform computational models of how, neural circuits work in the time to come. Now, the obvious limitation of expansion, of course, is that it doesn't work on living things. And so that's a nice segue into the second short story I want to tell you about, which is optogenetics. So optogenetics is a technology that allows you to control the brain with light. This started when my collaborator Carl Dijsselroth and I were both students at Stanford, and we were just brainstorming, how do we control brain cells very precisely? And so we just started going through all the laws of physics. There's only so many kinds of energy you could deliver to the brain, and we thought light would be cool. You could bring light into the brain using optical fibers, in the same way that electrodes are commonly lowered into the living brain. Then the question became, how do we make brain cells sense light? Because most of them don't. And so I became really fascinated by microbial opsins. These are proteins that look like the one in the lower left. There's 7 transmembrane proteins found in single-celled microbes. And they're used by these single-celled microbes to convert light energy into chemical energy. So the first to be found was a light-driven proton pump. Later, people found light-driven chloride pumps. And later, people found light-driven ion channels. And just to catch people up to the present day, what we and our group members and alumni and collaborators and so forth have found is that basically all of these families of molecules can yield molecules that are safe, fast, and effective for controlling neural activity. Express the gene in a neuron, shine light, and you can turn neurons on or off. So by turning neurons on, you can figure out what kinds of, for example, behaviors they can initiate. You can figure out whether they can trigger a pathological or curative state potentially of the brain. By turning neurons off, you can figure out what they're needed for. Could you delete a memory or turn off an emotion, and so on and so forth. So many, again, just as with expansion microscopy, these tools are now in use by many hundreds, perhaps over 1,000 neuroscience groups. They're being used to study how activating or silencing neurons could trigger all manner of behaviors or pathological states. And so, in this short talk, we don't have time to discuss everything that's happened in the last, really almost 2 decades since our first paper on this, but I really just want to comment on a couple interesting recent developments. Last summer, a European team announced that they were actually starting to use optogenetics in people. So most of the results to date have been used in animal models like mice, to turn off a memory or activate an emotion or try to change, cancel out a Parkinson's symptom and so forth, which is great, right? We're learning a lot about the brain, which is very, very valuable. But a group in Europe did show that they could take a patient who has retinitis pigmentosa, a form of blindness that causes the light sensing cells of the eye to atrophy or die off. They put in a gene therapy vector with the gene for crimson, which is an optogenetic molecule that we reported in 2014. And this person had a partial restoration of functional vision. So this is a disease that can't be cured. And this person was able to recover partial functional vision, could recognize objects, could see lines like a crosswalk, couldn't read text or recognize faces yet, but still a major step forward. More commonly, people use these tools to study the brain. So my collaborator, Li Wei Sai, has used optogenetics to drive the brain of Alzheimer's model mice. These are mice that get Alzheimer's-like symptoms. And she showed that if you drive the brain 40 times a second, the brain gets better. The teams went on under her leadership to show that you could then simulate those brain waves by showing mice a flickering light through their eyes. No need for the optogenetics anymore. Just have the mice watch a movie, basically. And more recently, the team showed that you could add in a clicking sound, have the mice watch a flickering light and hear a clicking sound, and they will then get better. So now she and I have co-founded a company, Cognito, which is trying to, which is doing and has some very exciting early data showing that with actual Alzheimer's patients, perhaps exposing them to flickering lights and clicking sounds could be a potential treatment for Alzheimer's disease. And so it's early days. This is not an FDA-approved therapy. Neither is the blindness treatment that I mentioned earlier, but it points the way towards how optogenetics, either directly in people or by inspiring ways for modulating the brain to treat other diseases, could yield potential therapeutics down the road. And so the final short story I want to tell you about is, basically the opposite of optogenetics. Can we watch the brain in action? Now, with optogenetics, we were really lucky. The natural world made these molecules through evolution that allow us to control brain activity, basically without modification for the most part. But for imaging brain activity, we were not so lucky. You can't find a molecule just out of the natural world that all by itself will report, brain activity with high fidelity. And so we thought, well, if the world doesn't want to evolve this for us, let's evolve it in the lab. So we built a robot that does evolution in the laboratory. Many groups, of course, have done directed evolution before us, but we wanted to devise an evolution robot that would give us what we want: a fluorescent indicator, a glowing indicator of neural activity that would be safe, effective, fast, and so forth. And so flash forward to the current day, we use this robot to develop a molecule that we call Archon. And working with Shio Han's group at BU, we showed that the Archon molecule could be used to image neural activity with a fairly simple optical setup. Shine light on the brain from a red laser, collect the infrared light. What you see on this slide look like electrode traces, but they're not. They're being imaged on a camera. And what that means is you can image many cells, right? Because cameras have many pixels. Here's an example in an awake behaving mouse where we're imaging, 8 neurons and seeing activity in their brains. So we're very excited about each of these tool sets. We want to see the brain in action. That's being empowered by technologies like the voltage imaging that we're doing. We want to control the brain. That's where optogenetic tools can be quite powerful. And we want to map the brain. That's where expansion microscopy can be quite helpful. In these short form talks, I was asked to keep it to 20, 30 minutes. I didn't have time to acknowledge all of the people led the projects, but their names are on the slides. And I'll end on this slide because an even longer list of people in the lab at the top or collaborators around the world in the middle of the slide have helped to make these projects successful. And we share all of our tools as freely as possible. Please visit our website, synthneuro.org, in the upper right. And don't hesitate to email me at boyden@mit.edu if I can be helpful. I guess we have time for questions now.
Yeah, I think we still have quite a lot of time. Thank you very much, Ed. That was fantastic. As usual, a wonderful contribution. I'm sure we all have questions, even though it looks like we didn't all put them in the chat here. So I guess just speak up or put them in the chat now if you like. I'll I'll give it a start. I had one question that came to mind right away as you were talking about therapy for Alzheimer's patients. And I was wondering if you could say a little bit more about the sort of the mechanistic underpinnings of that. Like, what is the theory behind why this works or seems to work?
Yeah. So in the studies that Liwei and her group led, they have been investigating this, and we have some joint students working across their group and our group. Um, it does seem to clean up amyloid. It also seems to clean up tau. Blood flow seems to improve. Um, Microglia look like they're less inflammatory and more phagocytic. So a lot of different things change. And so what is the proximal cause, the thing that triggers everything else? I think, to be honest, we don't know for sure yet. It's still early days, but it's really one of the reasons that I was excited to, co-found the company and help get this into the clinic was it did seem to address many different problems. And that made at least suggested to me that, it'd have a better chance of being a shot on goal than a strategy that only addresses one of the many, many things that happen in Alzheimer's disease. So that's one thing I'm excited about.
Thank you.
I had a question, Dr. Boyden. My name's Andy. I'm a MD PhD student at Colorado. We met like a couple times, but I know you meet a lot of people. You've developed a lot of really interesting tools, even everything from like the wireless DBS to everything you talked about today. Do you think that we're at the phase now where we have enough tools to try to get at some of the deeper questions in cognitive neuroscience, or do we still need better tools like higher density recording probes or some type of angio recording method to get more network activity in live networks?
Probably depends on the scientific question, right? I mean, a lot of scientific questions can be answered with classic tools being used in clever ways. My first, when I was doing my PhD with Jennifer Raymond and Dick Chen at Stanford, my first paper from graduate school was pure behavior, just watching mice move their eyes. No molecules, no physiology, no imaging. And that was great training for me because I trained previously as an engineer. Learning how to really ask great questions, is a very powerful skill. And working for Jennifer and Dick was really great training. And yeah, looking at behavior is important, right? You can learn a lot by just looking at things. So yeah, I mean, in our group, we'd like to combine all these in small brains, like fish and worms. So the worm C. elegans has 302 neurons. The larval zebrafish, maybe 100,000 or so. A mouse, of course, has what, 100 million or so? And a human has on the order of 100 billions. So, so one of the interesting questions is, can we combine these techniques, whole brain imaging, expansion, optogenetics, and so forth, and integrate those data sets? So I think in the coming years, that's something that we really want to do. I would love to see if we could simulate entire small brains, for example.
Yeah.
I think that's a reasonably ambitious, but not entirely unwieldy goal, given the quality of the current tools. Will we, and this is what I sometimes like to call real science. We don't know what the answer will be. So the answer to many more of your questions might be, I don't know.
Fair enough. Thank you.
Hey, Edward Boyden. My name is Rashawn. I've actually known about you since I was 18 years old and since high school. I even wrote a blog post building off of some of your ideas. So it's quite amazing that I get to talk to you personally now, thanks to Allison here. But my question is, I guess half question, more of also statement. I feel like, have you heard of a woman named Lou Jepson?
Yes, we almost shared lab space when she was going to start MIT faculty herself. Yeah.
Oh, okay, great. Okay.
Up to do startup companies. So.
So of course, her research, because you mentioned that with brain imaging, her research, if you probably know, is would be groundbreaking, completely groundbreaking. If I guess if not everyone here knows about her, but basically she's building what is essentially a very high fidelity, high quality MRI in the size of a cap. Literally, you can put it on just your head. It's so small, and the actual physics behind it is very complex. But basically, she allows you to see at the level of a neuron in real time. And you can also see there's no limit in the depth as well. And I remember watching a talk, she knows about your work. Not only would this allow for reading, like if you like reading the brain, reading all the neurons in the brain, doesn't matter how deep you're looking as well, but also you could, with Edward Boyden's technology, if you combine the two, you could potentially write to the brain as well, read and write. And this would be basically this would be a non-invasive form of, very high quality, non-invasive form of what Neuralink is trying to accomplish. I'm curious if you, so you already know about her. Are you interested in actually doing something like that with the, I guess, the technology she and her company is building? Open Water, I believe, is the name of the company.
Yeah, always eager to learn about technology and try out technology. And yeah, as soon as there's something where we can look at the specifications and so forth, eager to learn more for sure.
You have heard about that particular helmet, or it's more of like brain imaging cap that they're building. Yeah, you've heard of this as well.
She and I have talked about it many times. Yes.
I see. Okay, good.
Yeah.
We also have Nicola with a question.
Yeah, sorry to ask a question that might pull us a little bit to the side. I'm very excited about the brain reconstruction. But my question is going to be different, just relative to the techniques that you described. That's just half of the techniques that you have developed. You have developed so many other techniques. And recently there was a talk about, Elon Musk and all those robots. And there's a huge level of fanciness about surgical robots that work on the brain. And I remember that at the time you were working on a robot that was supposed to do craniotomies for mice. And I know how precise this has to be and how many difficulties there are. So if you can just express some comments about how do you see robotics in neurosurgery in the future, and where you think this can go, and how many difficulties they will have to resolve before being able to do something meaningful?
Yeah, so we've had several robotics collaborations in the past, which yielded a autocraniotomy robot, a patch clamp robot, which is a way of introducing electrodes into the brain to record neurons. And yeah, those are now commercially available devices. And we once something is out there, we kind of move on to the next thing. So that's why I didn't talk about it. I was trying to emphasize more recent things that we're working on now. But yeah, those technologies are out there, and people can use them. To my knowledge, they're all being used only in animal models in basic neuroscience. I don't know if they're being used in clinical realms, but I have not really followed that field quite as closely as much as those technologies have matured out of the lab. We still have a few projects related to those things, but mostly focusing on new topics.
I also have a question. One was related to the company that you said you recently started. What was it called? Corbido?
Cognito Therapeutics. That's doing the
Cognito Therapeutics.
trials.
Could you discuss a little bit more about what the roadmap of this company is and what's next for you there?
Yeah. So the company has described some of its early trials, and so that's out there. And Brent Bond is the CEO. He could tell I think your audience more about what the company's doing right now. But I think it's very exciting to see if the trials can be scaled up. And the ultimate goal is to go for FDA approval.
Very cool. Okay, I'll post more about it here in the chat. And then you also mentioned the kind of ambitious, perhaps not so ambitious goal of simulating small brain. Do you have any more, I guess any more like hunch to share of like what a potential timeline around this could look like, or what would even need to happen and as first steps to get there?
This is all very new. And we're exploring it actively, building collaborations, recruiting people to join the team. And yeah, I don't have much more to say at this point. As I mentioned earlier, this is what I sometimes call, real science. It's not like you follow the recipe and you're guaranteed pancakes or noodles at a certain time of day,
I know you just said that you don't have much more to say about that, but I am really curious because, of course, generating huge amounts of data, either by using electron microscopy or expansion microscopy or dynamic recordings of any kind, you end up with piles of data. And neuroscience is used to looking at that data in the way where you studied it in the context of an experiment, and then you make a hypothesis, and then you build a model, and then you make a model that then tries to predict something that you then test. But there really hasn't been that much work in taking data directly from this big pile of measured data and then trying to populate the parameters of a model based on that. I think that some of the work that for instance, Professor Lou Jepson is trying to do at Genelia right now to try to model visual neurons in Drosophila based on data that they actually acquired from electron microscopy is very interesting, but I don't see a huge plethora of that kind of work. And it feels to me like you get the best results from this kind of brain mapping science and all this technology that's being developed around it if you have a feedback loop, right? Where you're trying to use the data that's being collected to make something, to then see how well it works, how well it predicts, and then to go back and say, well, actually, we needed to measure this a little bit differently, or we really need that data, or something like that, right? You want to have this cycle going on. Where would you say is the sort of the best example of that kind of work that you know of, if you care to share?
Yeah, great question. I mean, I think Eve Martyr, of course, has done beautiful work where they, on the Crab somatic gastric ganglion, where they can record all the neurons, they know the wiring, they can look at the molecular content, and then they analyze the data from very first principles ways. They look at how does the gene expression, across cells correlate? How does what kind of parameters could generate a given output pattern? So I always learn a lot from her papers.
Yeah, that's a good example. Yeah.
I think Nicola also mentioned Sebastian Zion.
Yeah, that was an example. You all know Sebastian Sung's work. They spent lots of time, money, and involvement to rebuild a millimeter of human retina. And the outcome that he did was that he was able to use that structure to recapture a model that exists from the 60s about the function of the eye detecting moving images. And I think that the big problem between fixed data and data in motion, in dynamics, is it's the scale. It's always scaling it up.
Was the question how to scale up, or I didn't quite follow the end?
No, it was more of a comment. It's the scale. It's I mean the clarity technique. It's a great way to reveal structural properties of the brain. Transferring it into the dynamics of the brain, it's difficult. It's difficult because of scaling. It's difficult because of looking at different ensembles of elements interacting and acting.
Well, that's exactly why I sort of prefaced the talk by saying that we want to think of the temporal and spatial scales as the most important drivers of what we want, right? And that's why expansion microscopy lets you do scalable nanoimaging. It's why optogenetics and imaging and neural dynamics scale, because cameras and computers are always getting better and better. Yeah, so scale is really one of the prime drivers of the technologies we build.
Okay, wonderful. We have two folks lined up, Steve Potter and Andrew. Do you want to go first, Steve?
Okay, thanks. Fantastic stuff, Ed, as always.
Yeah,
I wanted you to talk a little bit more about the Somalarkon voltage-sensitive fluorescent protein. This has been a holy grail. I've been following this since Larry Cohen was doing his dyes, and to get a protein that has this single spike resolution, single cell resolution, millisecond resolution is fantastic. What are the limitations? Like, why isn't this? It seems like that would have been just like the revolution that would give us the most ground truth of what's going on in the brain of any technology I could think of, especially when you combine it with optogenetic stimulation of the neurons using the same optics that you're doing some recording with. So what are the current limitations, and why are we really taking off with this like you were with optogenetics when that came out?
Yeah. So first of all, it's just new, right? I mean, our first object paper was 2005. A lot of the discovery-oriented papers started really pouring out at a rapid rate, 2009, 2010, 2011. No technology exists in isolation. So we can build an indicator, but someone might need to, you might need a fast camera. You might need, an optical setup. There's a lot of moving parts. So I do think that there is a cycle beginning where, oh, you got a new indicator. Let's try to build a microscope that, optimally interfaces to it. Let's, but no technology exists in a vacuum. Second thing is that I think it is very clear that nobody's made a perfect voltage indicator. So as I was trying to allude to during the talk, with optogenetics, we're just so lucky that these molecules out of the wild, off the shelf, so to speak, were strong enough, fast enough, and safe enough that you could use them to drive and cancel neural activity. So for voltage indicators, Archon is a very high dynamic range, very good signal to noise, not as bright as some of the others, of course. There are lots of other molecules that are much brighter, but they have smaller dynamic range and lower signal to noise quite often. And then, so yeah, I think making a molecule that's bright, high dynamic range, fast, doesn't photo bleach, the perfect voltage indicator, I think you could argue, has not been invented yet. So we, if many others, are continuing to try to screen for molecules that are, fast, bright, high dynamic range, all these things at the same time.
Are these voltage indicators amenable to light sheet microscopy?
Potentially, I guess. I mean, the molecule will take light from whatever source and will then emit it. So light sheet, there's light field microscopy, all manner of point scan microscopy. Yeah, I think there's a bit of a mixing and matching that could be done where we have different indicators and different styles of imaging. It may also depend on what species one is looking at. Our group is working a lot on small animals like fish and worms, where if you have a light sheet microscope which has a fairly small volume that will fit between the two lenses, a small brain might be one that's preferred. But of course, there are large brains too that people work on, like mice and others. So it might depend on the exact scientific question at hand, to be honest.
You're nodding. Next one we have Andy.
Andy, your mic is muted.
Thanks for that. The answer to this is probably I don't know, or it depends. But if you were finishing your PhD around now and you were interested in building either therapeutic or commercial market products and tools, do you think that pursuing a more traditional academic path, professorship and grants and spinning out IP with your university, or going more straight towards a private route is best in the type of environment we're in, given the work that people like you have done to give us so many great tools and understandings?
So it kind of does depend, right? Because a PhD that was very clinical might have a different path than a PhD that was pure programming, right? A PhD that was very experimental and hands-on might be different from one that was more theory-oriented.
Mhm.
And then, of course, there were different timescales, right? I mean, the time between our first optogenetics paper and the paper in Nature Medicine last summer that showed the first data in people was what, almost 17 years? So that's probably longer than a dilutive capital funded startup would probably be able to tolerate. And so on the other hand, when the project that I mentioned that Liwei led on movies for Alzheimer's, um, you know, doing a scaled human trial, is something that could be envisioned on a reasonably short time scale, right? So, yeah, it probably depends on the nature of what the PhD is about, what the nature of the problem is. Yeah. Did you have something specific in mind?
Yeah, so I do in vivo electrophysiology. I work pretty closely with neurosurgical teams. You're one of your ventures, like the inner cosmos idea that you and a few others were working on, like some sort of neuromodulatory approach, leveraging things like wireless DBS and TMS and maybe near infrared recordings, like that type of thing. I don't know. I guess, yeah, that's the route. That's my background. That's the skill set I have and what I'm interested in. And I don't know, it seems pretty even in my mind which route would be most advantageous. But yeah.
Yeah. Well, the other thing too is that new skill sets can be acquired, right? And as we discussed earlier, some things like in vivo physiology could be automated, so that the skill required to do it would be far less. So the skills are always evolving, and the technology is always evolving. Yeah. So it kind of depends, whether one is sort of driven by a kind of impact, right? You want to solve this problem, and if you just switch fields and become a chemist, that's what you do. Or do you like a certain skill? Some people just like to write code. Are they going to do it for Amazon or Google or for a startup or for an academic group? That's negotiable. So it depends if you're skill driven or impact driven.
Yeah, thank you.
Wonderful. We had another question, I think, from Archon. Do you want to ask it yourself? Or?
Yes, I just wanted to know if Boyden has heard about bioelectric computation outside of the nervous system. Research by Professor Michael Levin from the Allen Discovery Center at the Tufts University. It's quite very recent, but I would say completely groundbreaking research when it comes to, well, biology and generally, a lot of different things. But yeah, I'm just curious. Have you heard about this?
Oh, absolutely, yeah. And his group has used some of our optogenetic tools to drive electrical functions. They use one of them to drive regeneration of a tadpole tail, if I recall.
Yes, yeah, exactly.
They used the molecule we published in 2010 to do that. And yeah, there are lots of computations that occur without brain cells. I mean, even beyond electricity, cells in the body have thousands of gene products, and they're sensing and they're acting. I mean, our immune system is a great example, right? It can detect an invader, it can fight it, it has memory, right? It remembers the invader, so it can fight it better the next time. That's how vaccines work and so forth. And people even have mapped out some of the pathways in immune cells, and they have very interesting relationships to the pathways that are involved with human or neural computations of different kinds. So yeah, computation is ubiquitous in biology, right? If you think of it as a way of taking some kind of input, making some kind of evaluation of what to do, and then making some kind of output. And I think it boils down to the simple problem that all living things have, but is that your cells or your brain can anticipate many outcomes, but in the end, your body has to do approximately one or a few things, right? I can't be out finding food and finding water and avoiding the lion and everything all at the same time. At least it sounds like a stressful day if I'm doing all that. So yeah, I think computation is ubiquitous, and Michael's work is a great example of that. Yeah, lots of people now are finding that very simple organisms can do extremely sophisticated things.
Yeah, they showed I found it so amazing when he didn't necessarily show this, but this is one example. When he talked about how the butterfly, well, when it first is a caterpillar, it will actually, even though when it's transforming into a butterfly, its body turns into basically indifferentiated goop. There's no brain cells, there's no skin cells, there's no body cells. It's all indifferentiated as it then turns into a butterfly. But it will actually remember things that it learned when it was a caterpillar after it turns into a butterfly. It's amazing. It shows that memories don't even need to exist within the connections between the neurons. It's just completely, and of course they show other things as well, like the fact that you can actually see like a bioelectric outline of a face of the tadpole before the face even forms. Like that completely blew my mind.
I shared more info on Michael Levin's talk also in here for those of you guys who want to know more about it. We also have a few more folks in the queue. I think we have Steve and then Wendell again. Just watching the time. If not, 10 more minutes left. Steve, would you like to unmute?
No, thanks. I had just accidentally left my hand up. I'm enjoying listening.
Okay, then
Can we have Randall
I'll go ahead. Yeah, I just wanted to try to see if I could turn things around for a second because, you know, we've started this neuro group and we get together every once in a while, and Ed was so nice to come out and give us a talk and to field answers to all of our questions. I was wondering, what can we do for you? Like, what is it that makes you want to come talk at a group like this? What are you hoping that maybe you could get out of it? Is there something that anyone can help you with, or what is your biggest desire right now that you'd like to push forward, either in general or for your group or your lab, or is there anything like that?
Well, I'm a big believer in serendipity. You know, great discoveries are driven by luck. So we're always looking, you know, think about CRISPR, right? Think about PCR, think about penicillin. These are all stumbled across by basically by accident, right? The natural molecules, aspirin, right? Optogenetics comes from natural molecules. So yeah, I'm always trying to find out, you know, how do we build the team with the insights that will get the next revolution, you know? And so we're launching a network to try to collect organisms from all over the earth, which we can then analyze for new tools. Yeah, I'm always seeking serendipity.
Serendipity it is then. Okay, fantastic.
Can I comment on Randall's question? I think something that we could really use in this field is somebody like Carl Sagan, who could, you know, be a spokesperson to the lay public to get a little bit more acceptance and more excitement about it. You know, there are a few people out there who are trying to do this. David Eagleman is, and you know, Stephen Fry had a very good series on the brain. There wasn't a whole lot of this high-tech stuff, though, in it. I think if anybody here wants to become the spokesperson for this, I think that would be wonderful.
That's a good idea.
Right.
I mean, I think that Ed's doing quite a lot of that. At least whenever I ask someone, that your name always usually pops up as the first one that people feel really inspired by. So whether it's accidental or whether it's on purpose, I think it's happening to some extent. I think also one question that we always ask our speakers, which we send via email, is if there's one challenge that you think could really advance this field tremendously, then where you're always like, I just wish this one particular thing was solved, that you want to put out there for potential others to take on, what comes to mind?
Do you want me to describe a problem that needs solving? Is that what the question is?
Yeah, a problem that needs solving, where you like, if only this one particular thing was solved, you know, then all of that work in our area would be much easier. This can be something inside of your field, but it could also just be perhaps an adjacent field that just needs to progress so that you can make more progress using those tools. And we talked a bit about tools before, but I think we always want to just leave space for like, what's the number one thing that people in the field think needs to happen to drive the field forward.
Good question. The problem is we often don't know what we don't know. There's so many molecules in the brain that people characterize at the chemical level, but we don't know what they do at the systems level. For example, some brain cells will generate gases like nitric oxide. What are they doing in a specific behavior? Well, we don't have the tools to see or control nitric oxide in specific 3D locations throughout a brain in awake behaving animals, not to my knowledge anyway. Cannabinoids, right? Brain cells make molecules that are, have some of the same features of the active ingredients in marijuana. What are they doing? The list of things that we don't know is just so large. So, yeah, I think we need to see everything in the brain and control everything in the brain, I guess. And yeah, that's at a very early stage.
Wonderful. Thanks. Randa, I'm not sure if you'd like to close it out. You're welcome to.
No, you can go ahead and close it out. Sure. Yeah.
Okay. Well, thank you so much, Ed, for joining. This was really wonderful. I think really sparked a ton of excitement in that space. Thank you so much for being willing to join this group, and everyone else. We'll have our see and control everything in the brain. Sound a bit like fundamentals talk about weather. Yes, Anders. Yeah, for everyone else, we are looking forward to seeing you for the next talk in the series. Thank you so much, Ed, for your time. It was really wonderful. And yeah, I hope you all have a rest of a lovely day, and we'll see you very soon.